Good afternoon. Nice to see so much interest in TreeFrog and so many familiar faces in the crowd. For those who I don't know, my name is Daniel Castro. I'm the Chief Business Officer at TreeFrog Therapeutics. Thank you to the MESA, as well as the sponsors, for the invitation to come and tell you a little bit about TreeFrog, our underlying technology, as well as our therapeutic pipeline. So TreeFrog was founded in 2018 by our scientific co-founders, Kevin Alessandri and Max Fair. In founding TreeFrog, Kevin and Max developed a technology platform, now called CSTEM, to address several key challenges within the therapeutic space. The first was to create therapies using a differentiated format. And that format is a 3D microtissue, as opposed to cell therapies based on a single cell format. And we think that microtissue has several key advantages that I'll describe in subsequent slides. Additionally, we can use CSTEM to manufacture at scale while maintaining cell quality. According to clinicaltrials.gov, there are currently 1,500 ongoing clinical trials. Not surprisingly, 90% of these are in phase 1 and phase 2. And for each of those cell therapies, those patients, each patient will require hundreds of thousands to tens of billions of cells. As those therapies continue to progress towards larger, more robust, later stage clinical trials, and hopefully we see additional approvals, as an industry, we will need platforms to be able to manufacture at scale and, again, maintain cell quality. We think CSTEM meets that challenge. CSTEM now has 120 employees that we affectionately refer to as froggies. Our expertise ranges between physics, biologists, as well as engineers. We have 3 sites. Our headquarters is in Bordeaux, France. We have lab space in Kobe, Japan. And then we also have a hub in Lab Central in Kendall Square in Cambridge, Massachusetts, which is where I am based. To date, we've raised $82 million from investors that include BPI France, Leonard Green, as well as Bristol Myers Squibb. Okay. So as I said, we are using CSTEM to build out our internal therapeutic pipeline. Our lead indication is in Parkinson's disease. We expect to be in the clinic by 2025. As this audience well knows, Parkinson's is characterized by the progressive degeneration of DA neurons. These neurons are responsible for the production of dopamine in the central nervous system. In Parkinson's, you typically start to see symptoms appear when 90% of these neurons are lost. Parkinson's remains a worldwide unmet need. There are greater than 6 million patients worldwide. In the US, total annual costs exceed $52 billion, and currently there's no disease-modifying treatments available, although several iPSC-derived treatments are in early-stage clinical trials. Importantly for us, Parkinson's has 30 years of scientific rationale and scientific research for cell replacement strategies to treat Parkinson's disease, dating back to the 1990s with fetal grafts. So for us, Parkinson's was an obvious choice for a lead candidate, or for a lead indication. So this is a snapshot of our Parkinson's program. We have a robust and deep preclinical data set. In the interest of time today, I won't dive into it, but at least at a high level, as I've mentioned, we use 3D microtissues as the treatment for Parkinson's. Again, this is opposed to single cells that are grown in 2D. And we think that microtissue format is a differentiating factor for us and derives several key factors. The first is that this microtissue, based on preclinical data, are highly functional cells, as measured by dopamine release in, again, preclinical studies. Additionally, we see that this format, this microtissue, is a highly efficient format for grafting. We see enhanced survivability of these microtissues post-transplantation. And finally, because of the highly functional and the efficient grafting, we believe we're able to control dose with this. So fewer cells, higher safety profile, more reproducible results. On the right-hand side of the screen, you see just an exemplary data set from our Parkinson's program. So this data was generated in Heme Parkinsonian rodents. This is a standard in vivo functional assay for Parkinson's. And you see 4 or 5 months post-implantation with either cryo or fresh microtissue, we see functional recovery of these rodents. As we continue to advance our Parkinson's program, we continue to look for additional indications in therapeutic areas where we believe we can make a similar type of impact. C-stem was really designed to have the ability to generate microtissues across many different cell lines. So to date, we've generated microtissues, 6 different microtissues from iPSC-derived cell lines, as well as 3 donor-derived cell lines. In all, over the last 2 years, we have filed 17 patents. We remain excited by the broad applicability of C-stem across these many different cell types. Okay, so I've told you a little bit about what we're up to in terms of building a therapeutic pipeline. Let me tell you a little bit about how we're doing it. So this site captures the essence of C-stem. And again, in founding TreeFrog, Kevin and Max had the key insight that there's a need to mimic the in vivo microenvironment when creating these cell therapies and manufacturing these cell therapies in order to preserve cell functionality and quality. So we create these therapies in a 3D format. And on the cartoon on the left, you see an illustration of that format. So working from the inside out, in the middle, you see a 3D self-organized microtissue. This is the therapy. This is the type of format that we use in Parkinson's. Surrounding that is an extracellular matrix, and then surrounding that is the allogeneic capsule. And that capsule is really the magic behind C-stem. That capsule protects the cells from environmental factors, such as shear stress, while still allowing for the diffusion of nutrients. And it's that capsule that allows us to, with a single capsule, what happens in that single capsule at a small scale is the same thing that can happen with tens of billions of capsules at a large scale. So this allows us to efficiently scale up manufacturing. If the video works, on the right-hand side of the screen, you see a video from our in vitro platform. Let's take a look at that. Good. In this video is demonstrating the self-organization and amplification of cells on our platform. And you'll see the video highlight the same features that I just illustrated in the cartoon on the left. And so these cells are able to replicate in an almost in vivo-like environment that replicates the human embryonic environment. Okay, so I had zoomed in a bit on C-stem and showed you an individual capsule. Let me zoom out and tell you a bit about how C-stem is implemented. So in the middle of the screen, you see a representation of our C-stem platform. This is a microfluidic platform. It's high throughput. We can generate over 1,000 capsules a second. And so we're able to feed the hydrogel, the stem cells, the matrix into that microfluidic platform. And there the cells are then encapsulated. Once the cells are encapsulated, they can follow one of 2 different avenues. On the left-hand side of the screen, the cells are seeded into up to a 10-liter bioreactor for expansion. So they're seeded into that bioreactor, and we're agnostic in terms of the type of bioreactor. At the end of the 7-day process, those cells the capsules are disassociated. Very, very simple process using a calcium chelator that takes mere seconds. The cells are disassociated, and then those cells can be used for either banking or they can be reintroduced for further expansion into the C-stem platform. Alternatively, the cells can go to the right-hand side of this graph, where again, they're seeded into up to a 10-liter bioreactor for expansion and differentiation. So here, they're seeded into the bioreactor. They're allowed to expand and differentiate into your microtissue of choice. Then that microtissue is then ready for implementation into Parkinson's or whatever indication that you're currently pursuing. Okay. So as we progress towards the clinic in Parkinson's, we have implemented C-stem into a GMP-ready device. You see a picture of that device on this slide. This system we call our Sakura. This is a high-throughput, fully automated, GMP-compliant system. It's fully closed and uses single-use sets. The software is fully automated and it's 21 CRF Part 11 compliant. And again, this Sakura is being used in Bordeaux. We use this to generate data for our Parkinson's program. And again, importantly for us, it's high throughput. So it can generate over 1,000 capsules per second and can be used to seed up to a 10-liter bioreactor. Okay. And with the delivery of the Sakura system, we have been able to generate some impressive scale-up data. So I would draw your attention to the middle of the slide with the 4 different data sets. The first data set is at 30 mLs. This was done off of our R&D system. The 3 additional sets are at 500 milliliters, 1.5 liters, and 10 liters. Those last 3 data sets were generated off of our clinical system. And you can see excellent, consistent behavior and predictability across those platforms and across those scales. And again, thanks to the capsule, we are able to predict, that the small scale is able to predict the results at the large scale. At the large scale, we have unprecedented iPSC amplification. We are able to generate 276x amplification after 6.5 days, while maintaining high viability at 98% and robust pluripotency at 97%. Okay. And being the BD guy, I would be remiss if I didn't end on a partnering slide. So for us, we think we bring differentiated value to our partnerships. We think we are building best-in-class medicines using a differentiated format. We think we have a disruptive technology platform, C-STEM, that can manufacture to scale while maintaining cell quality. And for us, we think we have executed a number of innovative partnerships. So to date, we've executed several partnerships, a handful of which we have publicly announced. And in those partnerships, we're pairing C-STEM with the partner's therapeutic asset in order to speed the development of that therapeutic asset. And as we turn towards 2024, we'll be looking to in-license therapeutic assets to augment our internal therapeutic pipeline. And again, pair those assets with C-STEM to accelerate the development of those platforms, of those technologies. All right, so I will end there. Thank you very much for your time and attention. I think rather than take a rushed question or two here at the end, myself and several of my colleagues, including our co-founder, Kevin Alessandri, will make ourselves available in the back hallway if you have any questions. Thank you very much.